made a small molecule inhibitor, which is kind of cool because it could translate into the clinics. So the target we do is HUR. It's a very pro-tumor RNA regulator. So we've got a small molecule that inhibits it and might be useful in clinical trials, but also in neuroinflammation because its role there is pretty big too.
Yeah. So, Dr. King, we have started the live. So I think we'll wait for 5 more minutes, Shreya, and then we'll start. We'll just share the link so people start to join in.
Yeah. All right.
We're live.
So they what they see is a slide, and then
it's a slide. Yeah.
Me and the slide, right?
So that they will see when you start. So now it's just a slide.
Okay. Yeah. All right.
So I will do a small introduction, and then I will ask you to start, basically. Yeah.
Okay. Then you'll kind of give the screen over or something.
Yes, exactly.
Okay. Very good. How long you guys been doing this program?
It started from 5th of June. So, yeah.
So this is born in the COVID era, huh?
Absolutely.
Yeah. I've had 3 grandchildren born in the COVID era, one just this past week. So there's good things that are coming out of this mess, you know?
Yeah, at least some good things.
Yeah, right. Yeah, of course, in our country, we're facing a big resurgence of the virus in the last month, like a 3rd wave. How is it in Switzerland?
Oh, it's not good at all. The 2nd wave is really, really strong, and countries around us have gone into their 2nd phase of lockdown, and somehow Switzerland hasn't. So it's a bit unsure how things are going to go from here now.
Yeah, I saw that France is in a lockdown now.
Yeah.
And is Italy in a lockdown too, or?
No, but they have more stricter measures than what it is here. Germany is going into a 2nd lockdown as well.
Do you have a national mandate to wear masks?
Yes, that we do. And this people follow very strictly. So that's at least nice.
Yeah, therein lies the problem in our country.
I know.
You get these stubborn people, and it's all politicized and everything else.
Yeah, indeed.
It drives me crazy as a clinician.
Yeah, that's completely crazy. I don't know how this works.
Hopefully the vaccine will come out.
Yeah.
It's such a bizarre virus. My daughter, when she was pregnant, she got the virus and lost her smell and taste. Really wasn't sick. Maybe had a little headache. And her husband, didn't get it at all. And they're like, husband and wife. And so so why he didn't get it, and he hadn't had it before, so he didn't have antibodies necessarily. So it's just a bizarre kind of virus, and the epidemiology is kind of bizarre too. So.
So I think, Shreya, we can start in a few minutes. Shreya, you ready?
Yeah, I'm good.
Okay. So, Dr. King, we can start?
Sure.
Okay. So we'll go with the countdown, and then I'll put to Shreya, okay? And I'll put myself on mute.
Yeah. Hello, everyone. Thank you all for joining us today for the 21st edition of the Progress and Prospects in Biology seminar series. And thank you for continuously joining us over so many past weeks and keeping this platform alive and buzzing with content. I'm Shreya, and I will be your host for the day. And without further ado, let's welcome our speaker for the day, clinician and scientist, Dr. Peter King from the University of Alabama at Birmingham in the United States. Professor Peter King graduated with a BA in zoology from Duke University in 1981, and then an MD at the Duke University Medical School in 1985. Following this, he did his internship in internal medicine at Case Western Reserve University from 85 to 86, and a residency in neurology at Duke University from 87 to 90. He completed a molecular biology fellowship in Dr. Jack Keen's laboratory from 90 to 91. And then he moved on to a clinical neuromuscular fellowship at the Cleveland Clinic from 91 to 92. He is currently professor and vice chair of neurology at the University of Alabama at Birmingham and chief of neurology at the Birmingham Veterans Administration Medical Center. He is co-director of the Comprehensive Neuroscience Center at UAB as well. His research examines the post-transcriptional regulation of cytokine and chemokine expression in glial cells of the nervous system and their impact on many neurodegenerative disorders, including ALS, of which he will talk to us today, and as well as other diseases like glioblastoma. Dr. King's lab was a pioneer in the field, showing a role of the HU family RNA protein binding factors in cancer. And as if all these credentials aren't enough, Dr. King also has a passion for music and plays classical electric guitar. Professor Peter King, we're extremely happy to have you with us today and can't wait to hear more about your research. The floor is all yours.
Thank you very much. You can still see the slide, is that correct? So good morning, good afternoon, or good evening, depending where you are in the world. Thank you for having me give a little bit of an insight into this disease, ALS. This talk will kind of combine some clinical aspects as well as basic science aspects, which I hope will kind of broaden your exposure to the entire picture of ALS rather than just one view. Now, 30 minutes is not a very long time for a very complicated disease like ALS. So clearly you're going to have to skirt over some issues. And if you have questions at the end, I'm happy to answer those. So we'll go ahead and get started. Let's see. Okay. So the overall view of today's talk will be to give a little bit of clinical insight into the disease, its heterogeneity, and the challenges that we face, as well as the pathophysiology. And there's three main areas under that category: disease initiation, disease spread, and rate of disease progression. And all those are governed by various biological processes that control that. And ALS is still a big mystery, and there are many unknown questions or answers to questions that I think are critical to developing effective therapy for this disease. In the clinic, we face patients that do not have much option in terms of helping them. And I think that will become clear after this talk. And it'll also become clear that we need young scientists such as yourselves to use your intellect and your energy to try and solve these problems, because this disease is very devastating. So let's go on to the talk. So what is ALS? A brief overview. ALS is a disease of motor neurons. Motor neurons control the muscles of our body, the volitional muscles, and allows us to walk, to talk, to use our arms, to breathe. And so they're very critical to the function of our body. We as neurologists view the motor system as two levels. The upper motor neuron system starts in the primary motor cortex and travels down the spinal cord. And this is kind of a schematic of broken up of how I think of it functionally. But basically, the upper motor neuron travels down into the spinal cord, into the brainstem, and then synapses into the anterior horn of the spinal cord at the different levels of the spinal cord. And synapse on anterior horn cells, which are basically the motor neurons that go out into the periphery and regulate the function of skeletal muscle. They do this by through the neuromuscular junction, which is where there's an electrochemical gradient that transmits the signal to the muscle and then permits the muscle to contract. So at every level, we as neurologists think of 4 levels. We think of the lumbar level controlling the leg. We think of the thoracic controlling the abdominal muscles and the erector muscles of the spine, and the cervical level controlling the arm and the hand muscles, and then the bulbar level, which controls functions of swallowing and speech. ALS, this is one of the heterogeneous aspects of ALS is its clinical presentation, and it can hit any part of the motor system in any one. So 75% will hit various parts of the spinal cord or levels of the spinal cord or spinal system, and then about 25% will begin in the bulbar or speech and swallowing dysfunction. The hallmark findings of ALS clinically when we see the patients in the clinic is basically atrophy and fasciculations. And I try to show that with these snapshots. Here's atrophy of the hand. You can see the muscle here, the first dorsal interosseous muscle is sunken out, and that's a hallmark of atrophy. And then here in a patient with bulbar ALS, they have severe atrophy of the tongue. So depending on the level of involvement of the disease, that the respective muscles become atrophied. When the upper motor neuron, which is the one I mentioned starting in the cortex, that gets involved in ALS as well. That presents a little bit differently and presents as increased tone in the muscle and spasticity. So those are the main clinical manifestations. The average age of presentation for this disease is about 60 years old. Slight preponderance of males, about 1.3 to 1. It knows no boundaries in terms of ethnicity or race. One other thing about the disease then clinically is that demonstrates its heterogeneity is the clinical decline and rate of decline. So in the clinic, we use a 48-point functional rating scale. It's very simple. It's not sophisticated, not a lot of science in this. It's basically activities of daily living, measuring those as the patient responds to a questionnaire. It's reproducible and reliable, so it does give good data, but it's not rocket science. And this 48-point scale, 48 being normal and 0 being basically fatal, most patients present, basically close to normal. But what's interesting about this graph, and the x-axis is time from presentation in months. So as you can see, there's a wide variety of decline rates. Some patients, don't decline at all for 6-7 years and all of a sudden plummet. Other patients present and just plummet within the first year and die. And so that heterogeneity of progression is a major question that we ask in the research laboratory as well as in clinical trials, because it can really interfere with our interpretation of responses to medication. And over here is kind of reflecting this. This was a single clinic right here experience, but this is an aggregate of all the ALS studies that a lot of the ALS studies that have been done, just aggregating all the normal data, normal quote unquote data in the natural progression of the disease. And this is the same axis. This is the functional rating scale, and this is time. And as you can see in these groups of patients up here, the slope is very small, and so they are progressing very slowly. And then you get to a group that's kind of intermediate, and then you get a group that's very quickly progressing. And so one of our challenges in this disease is to trying to come up with biomarkers that can help us to predict and subclassify these patients. And then lastly, about 2 more things about the clinical aspect. There's heterogeneity of survival. The younger you are, the longer you'll live, and that's due usually to the lack of comorbidities that we see in older patients. So those that are over 65 really have about a 3 to 4 year lifespan after the presentation of the symptoms, versus the younger patients that can go up 5 to 7 years. And then in between, younger being less than 45 years of age. This is a study we published a few years ago. It's a large retrospective study of our experience at UAB. The interesting aspect of this study is that we have a large population of African Americans in Alabama versus other states in the country. So we were able to look at, this is probably one of the largest African American studies. It's retrospective, but we found that there was no difference in survival between Caucasians and African Americans in either of these age groups. So again, ALS knows no ethnic or racial boundaries. One brief thing, this is an MRI. This kind of highlights the dual involvement of the motor systems, the upper motor nervous system that originates in the frontal cortex. And as you can see down here, the corticospinal tract fibers. This is a T2 weighted MRI scan. You can see these lighting up. That's abnormal. The arrows are pointing that out. And then if you come down into the spinal cord, into the anterior part where the anterior horns are, you can see these two eyes. They look like eyes, and those are very focal dots of degeneration in the anterior horns. This is not common to see MRI abnormalities, but this particular case demonstrates the selectivity of this disease. So we'll go on and talk a little bit about ALS pathophysiology and some of the basic research aspects of that. And we'll talk about disease initiation, disease spread, and then the rate of progression. And I think I've alluded to that in the clinical aspect part of this talk, that these can be quite heterogeneous. The way I think of disease initiation, this is a very simplistic model, but I think it summarizes a lot of what everyone believes, that there are multiple inputs and multiple different triggers that can initiate motor neuron disease or ALS. And it's probably like anything else, like cancer and other diseases, it's a combination of environmental exposure and genetic makeup. And I view these as initiators. They trigger the disease. And once the disease is triggered, they kind of all funnel down into the same final pathway, which is variable based on accelerators. And we'll go kind of separate these two in this talk, and beginning with the initiators to kind of go into that. So let's see. The obvious initiators here are genetic. And ALS, about 10% of patients with ALS have a gene that can be identified, or a genetic pattern in the family. And to give you an idea of the heterogeneity here, there's been over 30 different genes, and probably up higher than that as of last count, that have been linked to initiation of ALS. And these cover all sorts of biological pathways, again, underscoring the heterogeneity of the initiators. But what has come to light in the last few years is a somewhat common theme of RNA metabolism and RNA processing. And so this red circle genes here are all involved in RNA processing. Now, other ones are involved in other biological pathways, but as I'll talk about a little bit later on, most ALS scientists believe that dysfunction of RNA metabolism and processing plays a major role in disease initiation. However, you have to imagine, when you have this kind of a list of diseases, of genetic genes with multiple different pathways, that the hypotheses go wild, right? I mean, and so this next slide I'm going to show you, it's kind of hard to take for a Sunday morning, but the impact I just want to have you, so you understand where everything is all over the map. And these are the hypotheses for initiation. And I don't want anyone to read every individual, and I want the impact is just showing you the diversity of different theories. This is the muscles, this is the neuromuscular junction, this is the axon, and this is the cell body. And everywhere along the way, there are theories for initiation of the disease. When I was training many years ago, everyone focused on the cell body, and that has come to light. The RNA dysregulation that takes place in ALS occurs vis-a-vis RNA binding proteins that translocate from the nucleus to the cytoplasm. There's also proteasomal degradation abnormalities. There's mitochondrial abnormalities, aggregation of proteins that all can disrupt regular functions in the nucleus and the cytoplasm, many of which center around RNA metabolism. But then you go down into the axon and you can see dysfunction of axonal transport, either retrograde or anterograde. And even further down, in the last 5-10 years, there have been theories that maybe ALS originates in the periphery, and that the process is a dying back phenomenon. And there's the very first pathological changes that occur in ALS are at the neuromuscular junction, where there's retraction of the termini of the motor neurons, and there's also mitochondrial dysfunction in the skeletal muscles. So that has led people to believe that maybe the dying process goes the other way, rather than coming from up to down, down to up. So as you can see, it kind of creates a lot of confusion as to where do you target a therapy. And there have been therapies targeting all these different pathways, and I can tell you that most of them have not worked, and the ones that have worked are only modestly working. So what I would like to do is to kind of show what are some of the common denominators rather than all these different variable genetic presentations, because 90% of ALS is what we call sporadic, meaning there's no obvious genetic underlying cause. Now, I should preface it by saying that there's 30 genes have been identified. It's likely that there's the 90% that they don't identify these genes is due to just a lack of insight. And there's probably polygenic pathways that lead to the ALS, that you have to have polymorphisms or mutations in several different genes that have to align at the right time to create the disease. But that's kind of beyond the scope or our ability of bioinformatics these days. I imagine in the future, with the advancement of technology, that insight will be much greater. But for now, I think it's important to look at what is common among all the ALS patients, either sporadic or hereditary. And that is shown in this slide. First, I want to show here, this is kind of the hallmark neuropathological change that occurs in ALS. And these are the anterior horns right here. This is a normal control patient. This is a Luxol fast blue stain, so it stains myelin. And as you can see in the ALS patient down here, there's shrinkage of the anterior horns compared to here. And then there's loss of myelination or an atrophy of these corticospinal tracts. And these are the upper motor neurons that I mentioned that originate from the primary motor cortex. So it's a combination of upper motor neuron and lower motor neuron. And this just gives you an idea of the pathological changes. However, the one kind of common denominator that occurs in over 95 to 96% of patients, whether you have hereditary or sporadic ALS, is abnormalities of TDP-43. And TDP-43 is an RNA binding protein, and we'll talk about some of its biology in a second, but the pathological substrate that we see is basically aggregation of TDP-43 in the cytoplasm. So TDP-43 is normally a cycling nuclear cytoplasmic RNA binding protein, but it gets mislocalized or sequestered into the cytoplasm where it is aggregated. And you can see these big, like here, this big brown splotch here in the cytoplasm is an aggregation of TDP-43, and it can also extend off into the proximal axons in the hillock. We call these skein-like inclusions that are indicative of aggregation of TDP-43, and they also become phosphorylated and ubiquitinated. This same process occurs in the primary motor cortex, and these are cortical neurons where you also get aggregation of TDP-43. So both in the lower motor neuron system and the upper motor neuron system, you see this pathological feature. And like I said, it's in greater than 95% of all cases. And the TDP-43 is not mutated. This is wild type or normal protein. So in the next slide, it's kind of gives you a schematic of TDP-43. Like I said, it's actually an RNA and DNA binding protein that contains two RNA recognition motifs here and here. These are the parts of the protein that interface with with RNA substrates, and TDP-43 has a predilection to binding GU-rich sequences, but it has this interesting C-terminal domain, which is a low complexity called a prion-like domain, and it's glycine-rich, and this part of the protein allows TDP-43 to kind of self-aggregate normally in a cell, which occurs, for instance, during stress. It is part of stress granules, which are these transient organelles in the cell that come in response to stress and sequester RNAs and basically takes them out of circulation during that stressful process. And TDP-43 has this domain which kind of allows it to do this self-aggregation and then to disaggregate when the stress is over. Okay, so that likely predisposes it in a pathological state like ALS to continuous aggregation. And what initiates that aggregation or the oligomerization that precedes the aggregation is unclear. But it's a million-dollar question because this is a wild-type protein. It's not mutated. This particular slide indicates there are some ALS patients that have mutations in the C-terminal domain, but the vast majority is not mutated. So that's a real important question to try and understand the underlying basis for this disease. Nonetheless, when TDP-43 gets sequestered into the cytoplasm and kind of loses its nuclear presence, clearly that's going to have an impact on its nuclear function. And so that also provides clues into potential how it wreaks havoc on the motor neuron. So loss of nuclear TDP-43 is important in pre-mRNA splicing and microRNA processing. These are two nuclear functions. It's involved in nuclear transport of RNAs. So if it can't get back into the nucleus, it can't transport RNA. And then there's various cytoplasmic functions of TDP-43 that maybe get overrepresented when it gets mislocalized to the cytoplasm. So the real question is the dysfunction in RNA metabolism due to a loss of function in the nucleus or gain of function in the cytoplasm, or even just a loss of function overall? And as you can see, with the many different RNA regulatory pathways it controls, it can wreak havoc on on RNA regulation in general. And there have been multiple, miRNA-seq studies and RNA-seq studies indicating aberrant splicing that takes place when TDP-43 is knocked out, or if mutations are inserted that loses its nuclear localization signal that will trigger aberrant microRNA processing and splicing and other processes. So to continue on in the RNA theme, RNA dysfunction, I want to go to a hereditary form of ALS that was discovered a number of years ago that kind of is now the most common familial form of ALS, and it accounts for a high proportion. And this is C9orf72, and it accounts for up to 40% of familial patients. Mutant SOD1 is about 20%. So this is almost half of the patients that have a familial form have this mutation. And it's a hexameric repeat in the first intron of C9orf72. So this GGGCC hexamer basically repeats and can repeat up to 500 to 1,000 times, and creates this huge expansion of this intron. Interestingly, it gets transcribed, so that expansion gets transcribed into an RNA, to a pre-mRNA, and it can get translated. There's no methionine start site, so it's a repeat associated translation. So there are these latent signals in these hexamer repeats that can initiate the ribosomal machinery to produce proteins. Now, as you can imagine, when you have an iteration of this hexameric repeat, you get these dipeptides in various combinations, and depending on which direction you're translating, and it can translate in both directions. So you get these up to 6 different dipeptides that can wreak havoc on the cell, and this is how it can interfere with RNA metabolism. This was a study published a number of years ago that when this mutated or expanded CNRF72 is expressed in the cells, you get these abnormal RNA foci in the nucleus. So it's like aggregation of RNA, which leads to nucleolar stress and basically the manifestations of cell stress that are present in motor neurons. So it's a different angle, but the same theme of RNA dysregulation. It's unclear actually what RNA targets these are, but they clearly create a cellular stress that leads to apoptosis or to cell death. Interestingly, about 25% of another disease called frontotemporal dementia is associated with this as well. And this has brought light to the fact there's a spectrum between ALS and frontotemporal dementia. And it's beyond the scope here to talk about FTD, but that's of interest. So finally, the other angle to RNA metabolism is mutant SOD1, which is associated with ALS. It is a homodimer, superoxide dismutase, which is normally a free radical scavenger. But when you do 1 point mutation of this protein, it misfolds and creates a phenotype of ALS. And it used to be the most common one, but has subsequently been replaced by C9orf72. So we became interested in this because of VEGF, which is a vascular endothelial growth factor, is very important for motor neuron survival and neuroprotection. And we found in the ALS mouse model that there was a progressive loss, and this is a qPCR in the spinal cords of these mice, that a progressive loss of VEGF in the spinal cord over the course of the mouse's life, whereas in the wild-type mouse, VEGF RNA was preserved. And here you can see kind of a quantitative assessment of this progressive loss of mutated of VEGF. Which made us think that, again, a loss of a growth factor that's as promoting the ALS phenotype. So our lab has been interested in HUR, and HUR is an RNA binding protein that basically binds to AU-rich sequences, and this is a portion of the VEGF 3' UTR to which HUR binds, and it binds and it stops the degradation of VEGF. And it also promotes the association of a polysome to the RNA so that the protein can be translated. It's also cycling between the nucleus and the cytoplasm. It has all other functions, but in this particular case, it's basically stabilizing and promoting the translation of VEGF. Well, when we use this luciferase assay where we put on the VEGF 3' UTR on the end of a luciferase reporter, and then co-express that reporter in cells expressing mutant, 3 different forms of mutant ALS-associated mutant SOD1, we get a profound loss of luciferase activity, indicating that the presence of these AU-rich motifs and the presence of mutant SOD1 is leading to a loss of expression of this reporter. And so we then looked at mutant SOD1 and found that indeed mutant SOD1 acquires an RNA binding capacity, and that RNA binding capacity is toward AU-rich sequences that are present in VEGF. And so here's the VEGF 3' UTR with the AU-rich elements highlighted in gray. And this is a gel shift where we take radioactive probes from the 3' UTR containing those AU-rich regions and do a gel shift, basically putting in recombinant, either wild-type recombinant protein or mutated ALS-associated mutated recombinant protein. And we see a very prominent shift with the mutant SOD1, a very weak one with the wild-type. And interestingly, that is competed out when we add unlabeled AU-rich sequences. And so this kind of shows that here, just similar, very similar to HUR. So both of them can be competed out with cold AU polymers. And so it made us aware that the gain of function of mutant SOD1 was to bind to AU-rich elements in VEGF. Now, those are not unique to VEGF, and there are other AU-rich sequences involved in cytokines and other growth factors. So this is not the sole RNA that mutant SOD1 could bind to. And so here's actually a better demonstration of this AU-rich predilection. So here, this is the AU-rich polymer, and you can see that gel shift is basically blocked out, but it's not blocked out with a poly-C polymer. It is blocked out with poly-A. So AU and AU-rich sequences block this binding, just as it does with HUR. So it really acquires an RNA binding function like HUR. And it wreaks havoc on HUR's function. And so this is an example where we have an ectopically expressed vector that can produce mutant SOD1, 3 different mutants. And with those 3 different mutants, there's a profound loss of binding to HUR, and also to other RNA AU-rich binding proteins like TR1. So there's a competition that occurs because this mutant SOD acquires this RNA binding capacity. And if you look at polysomal analysis, which is basically we isolate polysomes through a gradient centrifugation, and you can isolate polysomes right here, which are in the later parts of the gradient, and then you can look at proteins present in there. And HUR typically is present in the polysomes because its function is to promote translation. So in a wild type SOD1, you get nice representation of HUR throughout the entire polysomal fraction. However, with the mutant ALS associated SOD1, you get loss of HUR in the polysomes. So that kind of helps us to explain the loss of VEGF, and potentially could contribute to ALS disease progression because of loss of that trophic factor. And so we also found that mutant SOD1 colocalizes with HUR, and it basically indicates that SOD1 can bind to, and this is basically an immunoprecipitation from the wild mutant mouse and the wild type mouse. And as you can see, there's co-immunoprecipitation of HUR and TR1, so it's not just limited to HUR. So the disruption of mutant SOD1 is probably extends beyond HUR, or definitely extends beyond HUR. And we also find that HUR gets sequestered into these aggregates, or form aggregates in cells that are expressing ALS-associated mutant SOD1. So it mimics a lot of the pathological aspects that we see in ALS. So we're going to talk a little bit about spread. This is briefly about spread, and then we'll go finish up on disease accelerators. So the spread of the disease is, interestingly, ALS begins very focally. And the first slide I showed begins in the hand, the arm, or the tongue muscles. It's almost reliably predictable in its spread. It starts to spread regionally from where it begins, and then over time it spreads up and down the neuroaxis. And you can follow that spread by TDP-43 pathology, those aggregates I showed you. So there is this theory of spread. Maybe it starts up in here in the primary motor neurons, or maybe it starts in the anterior horn cells, but wherever it starts, it then spreads in a very predictable pattern. And that spreading of TDP-43 pathology really indicates that there's some kind of signal, a degenerative signal that's doing this. And that's the million-dollar question, is what is that signal? Because as soon as we can find out that signal, and I think aggregation of TDP-43 is basically just the clue. I think that there's something underlying that initiates that aggregation. That spread, if we can stop that spread, because it starts focally, if we can therapeutically intervene, it would remain at that level. And that would be a very important intervention. So then disease accelerators, and those are clinically the slide I showed you where patients can progress really rapidly or slowly. We're beginning to realize that there are a number of modifiers here And this is, again, the kind of system we're talking about, that there's support cells around the motor neuron, both in the spinal cord, in the brain, and also peripherally in the nerve and the neuromuscular junction that can accelerate this disease. And so I'm going to just start off and focus the rest of the talk kind of on neuroinflammation. So here's an example of a study we did, and this is up in the spinal cord itself. We have a huge repository of human ALS samples that we've gotten over the years from our clinic. And here's the mouse. And this is basically the time course of the mouse, showing that the mouse basically develops ALS, loses its function, and expires. And toward the end stage of both patients and mice, you see this intense microglial activation. This is an IBA1 stained section of spinal cord. And you get this intense reactivity compared to wild type, where you not only is it intensely reactive, but they're amoeboid in shape. And so when microglia get activated, they kind of lose their projections and become amoeboid. And here you can kind of appreciate that amoeboid, those little spindly projections. So they're quiescent here and they're active here. So there's an active role of microglia. And the same happens in humans. And these are 2 ALS patients where you get this intense microglial activity and very little in control. And we think that microglial activation basically accelerates the death of the motor neuron, because these activated microglia secrete cytokines and chemokines that are toxic to the motor neurons, directly or indirectly. If you go down further into the neuroaxis, you see inflammation in the nerve roots, and this is at the level of the nerve roots. This is the mouse, and this is the human. So both nicely correlating, and this is CD68 and CSF1R receptor staining, and there's both of these macrophage markers are present in the peripheral nerves and roots of both the animal model and the patients. And then if you go even further down to the neuromuscular junction, you see an intense inflammatory reactivity. These red cells are basically neutrophils. This is elastase staining. They pick up neutrophils. These are neuromuscular junctions bundles right here. These little green donut-shaped things, and these are the gray shapes are the axons, the termini of the motor neurons. So they're ending up on these neuromuscular junctions. And as you can see, there are multiple inflammatory cells surrounding these. This is the early stages of the mouse. It progresses with advancement of paralysis in the rat model. And up here is an example of what happens over time is you get denervation of these neuromuscular junctions. And here you've got bungarotoxin and synapsin reflecting muscle and nerve respectively. They merge nicely, and here they don't merge at all, meaning that the motor neuron termini have withdrawn from the neuromuscular junction. And we see the same correlate in humans, and this is a confocal image of a human, one of our muscle specimens. And you can see this intense neutrophil activity surrounding this neuromuscular bundle right here, this junction right here. This is alpha bungarotoxin staining. So this inflammation is also extending to other cell types. So mast cells, which are another inflammatory cell, are also present in peripheral muscle and nerve. And this is, these mast cells, when they become activated, they degranulate and release all sorts of probably cytotoxic cytokines, as well as chemokines that draw in other immune cells. And they're spread all out. This is a skeletal muscle section from a human ALS patient, 2 patients, and a control. And you can see they're scattered all over the skeletal muscle. And then if you do quantitative assessment, it's significantly elevated compared to the controls. Likewise in the mouse, the rat model. And this is an example of where it's really important to have human and mouse tissue or animal tissue for correlation. And I think my role as a clinician is to be able to obtain that tissue and collaborate with basic scientists to help them correlate their findings in the animal model. Because at the end of the day, it's the humans that we're really trying to target and to help. And so I think that this is an example of collaboration and group effort to solve a problem, and not like a single cowboy. So finally, I'm going to end up at the level of the muscle. You see, again, these infiltrating inflammatory cells. And this shows you the pathology of ALS in the muscle, where you get atrophied fibers. And you can see right here, this is a group of angular, small, atrophic fibers, and next to fibers that are relatively normal. So this is called grouped atrophy, indicative of a loss of a motor neuron. And this is a biomarker study that I've been doing for probably about 8 years. And we did RNA sequencing and found that there's a real molecular signature that gets activated in ALS. And it's opened up so many different opportunities or directions for future study. And I'm not going to go into them, but this is some of the different markers that we identified through that RNA sequencing. And you can see here, this is the essential vitamin D activator. TGF-beta is a major cytokine that's important in fibrosis, and that gets activated in the ALS muscle. The Wnt signaling is activated here. So this is a Wnt antagonist that you can see here. Little red dots here. They're spread all throughout these atrophic fibers, and not really around the normal fibers of the muscle. And then concomitant with the FRZB is the beta-catenin, which is the major signal transducer for Wnt signaling. And it is intensely reactive in these ALS muscle samples in the endomysial connective tissue and along the muscle membrane borders. You can see this merge signal, yellow, is emerging between beta-catenin and WGA, which is a marker of muscle membranes. And then likewise, SMAD signaling is activated, and that has a whole other repertoire of regulation, including microRNAs and fibrosis-inducing gene pathways. And so this has opened up many avenues to determine whether A, these are involved in disease progression, B, whether they're therapeutic targets that we can then modify the disease progression rate, or maybe even they're part of the beginning of the disease. Like I said, this theory in the last 5 to 10 years is that the disease may start at the neuromuscular junction and then travel back up into the anterior horn cell. And so this leads us to therapeutic possibilities. And for the initiators, it's obvious that we can target, and probably in the near future with CRISPR technology, knocking out or repairing genes that are abnormal. But again, this is what only help 10% of the patients with ALS. 90% you can't, there's not an identified gene. The spread we talked about, anatomic spread, how we can stop whatever that signal is that's causing TDP-43 pathology to spread. That's a big black box. And then decelerators, that's what we're really relatively, most of the therapies now are aimed at decelerating the rate of progression of disease, but not reversing it. So these are the 2 FDA-approved drugs. The third one has just reached the New England Journal like a month ago, but their sole function is to slow the disease down. They do not reverse it. So, and to be honest, their effect is pretty modest, and they don't really slow it that much. So we are now entering a clinical trial that our preclinical trials that I was involved in really helped set the stage, and that's masitinib, and we're starting this trial at UAB in the next month. But this is to target the inflammation that I just showed you, the neutrophil and the macrophage infiltration of the peripheral nerves. And masitinib is the name of the drug, and it is a c-KIT inhibitor. But a tyrosine kinase receptor, it targets the tyrosine kinase receptor. And this modulates reactivity in mast cells and also in myeloid cells. And so when you treat the animal model with masitinib, you get this loss of infiltration of those neutrophils that I showed you, as well as mast cells. And in the animal model, there is a survival effect of the masitinib versus the vehicle treated. So with that, pre-animal or preclinical trial studies, we've been able to start this phase 1/2, which showed some slowing of disease progression. And now we're doing a phase 3 where it's an add-on to the already approved Ryliotech therapy. So again, though, this is just to slow the disease. And I think that that's the key thing. We really want to get something that stops it or reverses it. So in conclusion, I think that I hope that you got an appreciation for the heterogeneity of ALS, that there's multiple triggers for disease initiation, but once initiated, there's a common denominator, TDP43 pathology, that spreads within the neuroaxis, and it likely reflects a general dysregulation of RNA processing, and that the different areas of therapeutic targeting would be disease initiation, anatomic spread of disease, and then the rate of disease progression. And then I'll finish up 2 slides. Some of the research I present here is supported in my lab by the Veterans Association as well as the NIH. I've got great lab people listed right here. One that I'm sure you know pretty well, or some of you know. And then, as you know, research is a group effort and takes a lot of different people. And there's a lot of people that I work with in the ALS clinic that I direct that allows us to treat these patients, but also to enter them into clinical trials and to have opportunities to advance our research. So every paper I publish that includes those patients in the preclinical trials, the basic science trials, I acknowledge. And these are 2 papers we've published in the last year or 2. And I want to highlight to the group the importance of patients in contributing their tissues postmortem to help us. And if there's one thing about the ALS patient, it's a terrible disease. They end up, there's not much we can do to treat them, but they're some of the most generous patients that you can take care of. And they're all almost to 100% willing to contribute their tissues to help us advance our research. And as you can see briefly in some of the slides that I showed, how important it is to make that correlation between animal models and humans so that we can develop therapies to help patients with ALS. And I think I'll end there. And I'll take questions.
Thank you, Professor King, for such an amazing talk. It's really nice to hear about how a drug sort of goes from being in the laboratory to something that's clinical, or from the bench to the bedside, basically. And it's great news if a drug can be used to stop progression of such a devastating disease, such an ALS. In this context, I was just wondering, I will take questions, but I was just wondering if you can tell us a little bit, because this is so topical these days, the checkpoints that go into a drug from being in a preclinical trial to then a clinical trial, because this is not something we get to hear about much.
Yeah, so, I have a slide in one of my talks. It would take me a while to pull it up, but it's a great slide because it highlights the timeline for drug development, starting in the preclinical studies to getting in the clinic. And it's a very nice one because the timeline advances fairly well in the preclinical studies and even the safety studies. There's multiple levels that you have to do to prove that the drug is safe in the animal model. Then there's in vitro studies for humans as well that it has to pass. And then there's this big gap. We call it the valley of death, where most therapeutic modalities fall off into this valley of death. And it's due to the fact that when you advance it to humans, it's proven to be unsafe, or there's drug-drug interactions, or there's instability of the drug in the half-life is way too short, or it doesn't cross the blood-brain barrier for neurological disease. So there's many checkpoints that lead to that compound falling into this valley of death. And I think that the faster we can determine if that compound's going to fall into that valley, the better. And I think that's where we are improving, but it's still very laborious. I sit on a NIH study section that we review a lot of these preclinical compounds, and it's very costly. When you advance a drug from a mouse or some more primitive animal model to a mammalian model, like a monkey or whatever, that you can imagine the costs go up considerably. So, and the timeline also gets extended. So the checkpoints that are really kind of the hallmarks are: does it have favorable pharmacokinetics? Does it get to where it's supposed to go? Does it have target engagement? So that also implies that you need to have good biomarkers that can tell you whether your target's being engaged or whether you're seeing an effect, that you're expecting. And so I think that what people have to understand is that once you identify something in an animal, then you have to prove it to be safe in vitro, then in vivo, in more complex mammalian models, and then a phase 1 clinical trial where you just do a few patients and do a dose escalation, and then you just monitor about everything you can think of monitoring to see if it's going to be safe. And so a lot of drugs just fall off in any one of those steps. And so but nonetheless, it shouldn't discourage things from moving forward. I think in the one I showed you, the masitinib, that has already been being used in other disease processes. So it had gone through a lot of the regulatory steps. This is kind of a reuse of a drug, and that can really short circuit a lot of things. But we are now involved in a small molecule inhibitor we're developing for one of our RNA binding proteins, and we're at the very basic level. But we realize that we hope we can get it to at least a more complex mammalian trial before we retire. It takes upwards of 10 years, sometimes to get these compounds out. I'm not that young anymore, as you demonstrated in my bio. So,
well,
anyways, I know that's a kind of a disjointed answer, but it probably reflects the disjointed process that occurs. Yeah.
No, thank you for explaining that so well. I'll go to other questions from the audience. So there's a question from Jamie Darheny. I'm hoping I pronounce it correctly. So what should be the better approach to process lumbar spinal cord tissue for mass spectrometric analysis?
What should be the better approach? Well, we've already done that, and so mass spec analysis has really advanced quite a bit in terms of its tolerance for sample types. And so we just recently did a mass spec analysis of our ALS spinal cords. Basically what we did, we didn't do laser dissection of certain parts of the spinal cord, like the motor neurons, to get specific, but you could do that because the amount required for mass spec is very small. But we just prepared protein samples from our spinal cord sections and sent them off and got very nice mass spec results. Again, it's like an RNA-seq where you get a lot of targets. And the question is, and I'm going to kind of take a liberty to answer a little bit more. The problem is when you have so many different targets is how do you prioritize them? And of course, you do statistical filtering and you can get, kind of high priority ones, but then how do you validate it? And this is something I'm still arguing with the mass spec people because molecular biologists and, biologists such as ourselves and the mass spec people have different ideas about validating proteins in tissue samples. And so they laugh at me when I say, well, I tried to do this antibody. I did an immunoblot or I did, immunohistochemistry, and I didn't see the changes that you're showing. And they're going, well, you're looking at it a different way, in a very limited way. We're looking at it kind of three-dimensionally and in a more specific sequence-oriented way that the antibody may just not be able to recognize. And so I'm still at the stages of trying to figure out, because we got some really cool targets, but how to validate them. But I think the processing of the tissue is pretty easy.
Mm-hmm. There's another question from Jamie again, and there's a few questions about treatment of ALS. So the first one is, can immunotherapy be a treatment for ALS? And Is it now a treatment strategy already, or can it be in the near future?
That's a good question, and it's a loaded question because, I mean, I've been around long enough. When I was a fellow at Cleveland Clinic, we were involved in IVIG trials, immunoglobulin trials, which is a modality we use very often in neurological disease for autoimmune diseases and immune-mediated diseases. And that was my first experience, and it had no effect. And multiple trials with different immunomodulating medications, some of the typical ones like steroids, to azathioprine, to some of the more advanced biologics, don't seem to be very effective, and it's for unclear reasons. I think that the immune response in ALS is more complicated than just like, arthritis or something like that, where the immune response can be beneficial at some stages of the disease and detrimental at other stages of the disease. So, for example, in the ALS model, this is an animal study. It's a paper with, a few years came out showing that microglial activation early on was helped to, and that's vis-a-vis IL-10 production, which is, an M2-like cytokine, helped to delay the onset of the disease. Okay, so at early phases of the disease, microglial activation may be beneficial in slowing it down. It's a scavenger of debris, and we know that when cells are damaged in the brain, that is a potent stimulus for M1-like activation. So scavenging tissue debris and other contents may be beneficial early on, but then later on, I think it becomes overwhelming. The phenotype changes to more of an M1-like phenotype, and I think most people think toward the end stage that microglial activation is bad. But the problem is that at that point, a lot of the motor neurons are, a lot of them are dead or dying, and motor neurons don't regenerate. So if you bombard someone with immunosuppressive therapy toward the end stage, you're not going to really be doing much. Now, masitinib is intervening. It is an immunomodulatory drug. In this phase 3 study that we're entering in now is targeting patients that have only had the disease. There's a certain time limit for the patients to have disease, and they have to meet a certain functional rating before they can be entered. So we're not going to be targeting end-stage patients. We're not going to be targeting, so we're trying to give it the benefit of the doubt by starting early on. And I think that's another area of ALS that's of real interest, is to try and identify this disease early on. And I didn't go through the details of the clinical presentation, but it can be very challenging to diagnose. And the average time of diagnosis from onset of symptoms is 12 to 15 months. So, I mean, and usually patients come to you 12 to 15 months later and had 2 or 3 surgeries and various other misdiagnoses. And so we kind of get started late. And so if we can develop biomarkers that can identify early on the disease onset, it'd be of benefit for intervention. And I think, that the immune, that there's definitely a T-cell driven component to this. And I know Stanley Appel in Houston is a big proponent of that, and that by modulating, improving T-cell proportions, Tregs, that you can modify, at least in the animal, you can modify the disease course. But whether that'll translate into humans is unclear. Then there's also stem cell therapy, which is a very sexy title and grabs everyone's attention, but it's a very primitive level right now. And the idea there is for the stem cells to go in there and to form to differentiate into cells that can produce growth factors and support cells, but it's really not turned out to be very good. It's unfortunately because it grabs people's attention, it can be misused, and unfortunately, the ALS population is a vulnerable population, and they can be suckered into thinking they're going to get better and go off and get unregulated stem cell therapy at various places. So
I see. Thank you. Another question about therapeutic interventions, actually. So can antioxidant-mediated therapeutic interventions work for ALS? Because it's sort of known that the SOD deficiency leads to more oxidative and nitrosative stress in neurons. So can that be targeted somehow?
Yes. The recently approved edaravone, which is the trade name's Radicava, which was approved maybe 2 years ago, targets free radicals. It's a free radical scavenger that was initially used in stroke, but repurposed for ALS. And so it's a very vigorous protocol, and it is, of only modest benefit. I should say that, and I didn't go into detail, but mutant SOD1, the SOD1, if you mutate or delete both copies of SOD1, you get a little bit of abnormality, but you don't get the full-fledged ALS phenotype. So the thinking about SOD1 and the link to ALS is not so much a loss of function, but a gain of function. And I demonstrated that gain of function in terms of RNA binding, and gaining RNA binding function. But there's other areas where people have demonstrated gain of function. And so I think, but that doesn't downplay the role of potentially of free radical scavengers. And there are a couple other clinical trials targeting free radicals at various stages of progression. And so I think way I view ultimately treating ALS is it's going to be kind of like cancer, where it's going to be multimodality. That we're going to be targeting different pathways with cocktails of medication or treatments, not just one silver bullet that will just treat everything.
Sure. Yeah. So another question from Pramat Kumar Shai. How is sequestration or aggregation causing loss of TDP-43 function? What is the mechanism?
Well, I think that sequestration is an interesting question. So when these proteins aggregate, they TTB43 and other proteins, they kind of misfold, and when they misfold and get aggregated, certain parts of the proteins become exposed that are not normally exposed. SOD1 is a good example of that, but TDP43, same thing, that they can then sequester not just its own protein, but other proteins that might be electrostatically attracted to certain exposed residues, or hydrophobic residues can get exposed. So you might get other RNA binding proteins that have hydrophobic regions that interface with RNAs that get stuck into these aggregates, and it's and also RNAs, the molecules themselves can get stuck in there. And so the sequestration, leads to probably loss of other cellular proteins that will impair or impact function. Now, the interesting question, how does TDP43 aggregate? And I touched upon it briefly about the C-terminus and the low complexity domain, the glycine-rich domain, that allows it to self-aggregate and unaggregate, depending on the cellular stress. That there's clearly something that is preventing it from disaggregating or promoting its aggregation and not allowing it to disaggregate. And I think that there are other molecules floating around there that are interacting with TDP43, possibly RNAs, that are preventing it or keeping it in an aggregated state. And so, and the other thing is TDP43 cleaves in these aggregates, so the C-terminus gets cut off. There's some kind of protease activity that's occurring as well. So I think it's much more complex than we think, and there's a whole theory about liquid phase separation, that this protein can then separate from the rest of the cytoplasmic contents based on its conformation. And when that happens, again, other things can be attracted to it. Hydrophobic targets can be attracted to it, and who knows what gets stuck onto those aggregates, or which promote that aggregation.
Right. Thank you for that answer. The next question is from Shonak Shahu. He says, does the repeat length of the C9 ORF72 play a role in disease onset or progression? And what downstream targets does the RNA foci observed in C9 ORF72 affect?
Yes, the number of repeats does impact the phenotype, both for ALS and frontotemporal dementia. I'm not as familiar with the C9orf story, so I don't know specifically downstream targets that it affects. I think the RNA, there are these G quadruplexes that are formed with that repeat hexamer repeat RNA, and these G quadruplexes are these tertiary or secondary and tertiary structures of the RNAs that kind of also, lead to sequestration. And in terms of what specific RNA targets that get sequestered by these quadruplexes, I'm not exactly sure, but I think that it might be even dynamic and change depending on the cell, the state of the cell and the stress of the cell. But I think the main thing is that it does lead to sequestration of RNA foci and probably also, again, proteins as well. So I think everyone, the SOD data that I showed was a real eye-opener for me, indicating that any protein can become an RNA binding protein. And I think vice versa, that any RNA, if it's structured properly, can bind to a protein. And I think that the best example of that are these viroid RNAs that in tobacco and plants, these mosaic viruses, that these circular RNAs that are just 300, 400 nucleotides long. They don't have any coat proteins on them. They're just bare RNA, circularized RNA. They form these odd secondary and tertiary structures, and they wreak havoc on cellular proteins. And this is in the plant, not humans, but it disrupts all sorts of protein functions. I think, I can't imagine that in a correlate doesn't exist here in the humans or in mammalians.
Interesting. I see. I have a few other questions. If you have the time, can we go ahead, think?
Sure.
Okay. So Dr. Tonmai Sharnal asks, is there any probability to find out in case of whether ALS and Huntington's disease can be combined in a single individual patient?
Wow, that would be a double whammy. That would be really bad luck. I have not seen that. Now, the one thing though that can happen concomitantly is ALS and frontotemporal dementia.
Really? Okay.
That is a really interesting genetic phenomenon, and with the C9orf72 can cause at one end of the spectrum ALS and the other end of the spectrum frontotemporal dementia. But in families, some of them develop both. They get both FTD and ALS, and other family members only develop one or the other. So there's clearly modifiers that modify the phenotype of these genotypes, right? And whether that's some environmental exposure or some gender-related thing or some other environmental exposure, who knows? But I think that speaks of the complexity here. But I have not personally seen, someone with both Huntington's and ALS. But it's possible, but probably unlikely. Because most of Huntington's patients, the disease onsets in the 40s, and they usually expire, pretty young. And ALS is more of a older disease, although not exclusively, but typically 60 years old as the age of presentation. Now, the familial ones do present a little earlier. Yeah.
See. There's another question from Pranab Royce asking, is there any similarity in the aggregation of synuclein protein in Alzheimer's with this TDP43 aggregation in ALS?
Yeah, I associate synuclein aggregation more with Parkinson's disease, and in the substantia nigra, the pars compacta, the dopaminergic neurons develop these synuclein aggregates that then spread. I think there are commonalities that they begin with oligomers and these fibrils, and these fibrils then aggregate into larger aggregates. And there is also evidence that both can spread. And I didn't go into detail there, but TDP-43 aggregates like synuclein can spread from cell to cell. There's literature data out there that indicate maybe they get secreted by exosomes, vis-a-vis exosomes that then get taken up by a neighboring cell, and then can induce aggregation in that neighboring cell. And that is one of the theories for spread of ALS and for Parkinson's. In ALS, the spread is a little more complicated because you have a lot of geographic distance between the brain, the motor cortex up in the brain, and the spinal cord, right? It's, we're talking about like a couple feet maybe, and if you're short, maybe less. But in Parkinson's disease, the striatal dopaminergic system is relatively close, and you've got the striatum and the substantia nigra regionally very close, and so spread. Maybe it does spread from cell to cell, but you can't explain, all of a sudden having something up at the top of your head be present in the middle of your spinal cord. And that's what people think are focusing on other mechanisms of spread, and that's by way of the neuron itself, so that these aggregates maybe get transported, or the signal that causes the aggregation gets transported down the neuron to the synapse and gets synaptically transmitted to the next neuron. And that would be much faster, right? Because, you just has to go, it just travels down the neuron rather than kind of diffusing through the extracellular space, which, unless it gets circulated in the spinal fluid, it'd probably take a while for it to spread, much longer than we usually see clinically.
Thank you. I'll take two more questions for shortage of time. One of them is that, so you said ALS is prevalent for Caucasians, and it has a little bit of a lower rate in other types of people. So is there an environmental cause for this, or would you speculate some other cause behind this?
Yeah, that epidemiology studies in ALS are really problematic. There was, for instance, there was an island off of Japan where there was a hotspot identified of ALS, and everyone was thinking, is this something related to the ethnicity? Was it related to an environmental exposure on that particular island? I'm blanking on the name of the island. But there was a high incidence of ALS on that island. And so this was back in the 60s and 70s, 1960s and 70s. And when I was training, I wrote a chapter on ALS in the 1980s, and I reported on that. And I was thinking, wow, what could it be? Is it something they eat, or is it something they're exposed to? Is it their ethnicity? What is it? Well, lo and behold, when C9orf72 was discovered, 10 years ago, they went on that island, and it was all C9orf72 they had. And that explained this epidemiological blip. And that's a great example of how we can be misled by ethnicity or by environmental exposure when it's an underlying genetic problem. And with that said, my feeling is that the rates are fairly similar across ethnicities. I know that there's papers out there that suggest that African Americans have a lower rate, and that maybe Asians have a lower rate as well. But it's all very soft science as far as I'm concerned.
Mm-hmm. Nice. I will combine sort of two other questions. One is that Junisha asks whether ALS and MS, I'm assuming it means multiple sclerosis, they sound similar in a few symptoms. So what's the basic difference between the processes of these two? This is one. And the second is whether ALS and Duchenne's muscular dystrophy are somehow linked, and whether there's a difference in the genetic inheritance of these two diseases.
Okay, so the first question, drastically different diseases. They both have sclerosis in them. I guess that's the common thing, but the demographics are different, the disease course is different, and the treatment is different. So multiple sclerosis is a demyelinating disease of the central nervous system, and you get these inflammatory demyelinating plaques that can happen anywhere in the central nervous system, in the spinal cord, in the brainstem. And these are inflammatory lesions of the myelin and induced by the immune system, and nowadays readily treated by a lot of these biologics, beginning early in the 90s with beta interferons, but now has expanded. There's now up to like 14 different biologics that target different aspects of the immune system that block plaque development. Versus ALS, where it's a disease of motor neurons selectively, not totally selectively, but it is a progressive disease. Some would say that MS could be considered a degenerative disease because there is some progression, but it's not typically fatal. It can be fatal in some subsets, but usually these patients can live quite a long time with MS versus ALS, where they usually die within 3 to 5 years. The second question, let's see, Duchenne's and ALS. Well, Duchenne, it's a totally different genotype. It's X-linked. The dystrophin gene is the culprit here. There's variations of Duchenne's based on what type of mutation is occurring in Duchenne's in the dystrophin gene. Some of them are complete loss of expression of dystrophin. That's when you get the Duchenne's, and the boys, very young in age, start to develop muscle dystrophy and then usually die, in the early 20s. There's Becker's dystrophy, which is a more conserved read-through of dystrophin, so you get some expression, and the phenotype's not as bad. But in our lab, I have a junior faculty that's studying Duchenne's dystrophy. And he came to my lab, oddly enough, because I identified SMADs as a biomarker of ALS and skeletal muscle, and published a number of papers on that. And SMAD signaling seems to be important in muscular dystrophy. So there may be a commonality there at the level of the muscle. But that's like ground zero, and we're still not quite sure how that will be, but it's very interesting. I've learned a lot about muscular dystrophy and Duchenne's in the last 2 years, and and some of the overlaps at the level of the muscle. And like, for instance, TGF-beta is a cytokine that drives muscle fibrosis and muscular dystrophy, Duchenne's. It's also highly expressed in ALS muscle, and fibrosis is associated with ALS, fibrosis of the muscle. So there are parallels, but they're entirely different in terms of the underlying cause.
Thank you so much for that wonderful explanation. And thank you as well for joining us today. It's been such a pleasure to have you. And
Sure.
It was an extremely enlightening session, both for me and as well, I hope, for the audience.
Good. Well, good luck to everybody, and keep in science. We need to have people stay in science to keep us all alive.
That's true for sure.
Right?
That's great advice indeed. Yes.
Okay. All right. Well, I'll sign off then.
Thank you. Thank you so much.
Bye-bye.
Bye. So tune in to our channel next week as well for an equally stimulating session. And please don't forget to subscribe to our channel by clicking the bell icon to get direct updates. So we have 2 amazing sessions lined up for you next week. And please spread the word about our channel and help us share our network with everyone else. And see you next week. Thank you and have a nice weekend.